Hi, my name is Gabriela Rangel Cardozo, and today I'm going to be talking about cholera. I wanted to go more in depth about the path of the infection, so here you go. An overview of what I'm going to be talking about during this presentation is a bit of a background on cholera, as well as the infection process of the Vibrio cholerae. I wanted to talk more about the cholera toxin and what it really actually is. And I also want to talk about what positively and negatively affects it. So whether it's neutralizing cholera toxin or inducing virulence gene expression. A bit of background on cholera is that Vibrio cholerae is what causes cholera. It's the bacteria that causes cholera. It's a gram-negative curved rod with a single polar flagellum. You could see that right here. This makes it very mobile and it moves around a lot. It's capable of both respiratory and fermentative metabolism, which means it doesn't need oxygen to metabolize. It also secretes cholera toxins or collagen, and this is the reason for the symptoms that you see in cholera. The main way of transmission is fecal to oral. An example of this is consuming water or food that has been infected by infected fecal matter. To the left, you can see that there is a picture of the affected areas of cholera epidemics. The patterns that you see are usually common in developing countries, such as Africa and Southeast Asia. You could see that there are a lot of countries in Africa that have the epidemic, and Southeast Asia, as well as Australia and Mexico. Cholera toxin is what Vibrio cholerae produces. More specifically, it is produced by the O1 or O139 serogroups of the Vibrio cholerae O polysaccharide side chain. So there are many serogroups, but those two groups are the main groups that produce the cholera toxin. Cholera toxin is also made up of different subunits. There's subunit A and there's also subunit B. Subunit A is what is basically in charge of doing the damage, I guess. It is what does the enzymatic activity of toxin. Subunit B is what is in charge of binding the receptors of the host cell. So it binds to GM1 ganglioside, and that is the main receptor for cholera toxin. Some data show that the cholera toxin can enhance metabolism in Vibrio cholerae, and you can see this in the study that focused on doing that exact experiment. So what they did is that this specific study used rabbit pups that were infected with either wild-type Vibrio cholerae, a mutant, or a mock solution. The mutant was unable to produce either A or B subunits of cholera toxin. This means that the rabbit pup that was infected with the mutant wouldn't show the same symptoms that a wild type infected rabbit pup would show, which is the regular symptoms of cholera. The goal of this experiment was to see how much hemin was found in the ileum or the cecum of these pups, according to what they were infected with. So the hypothesis was if the rabbit pup was infected with the wild type, that there would be more hemin found. And you could see that this was true, seeing that the wild type rabbit pup had a lot more hemin found, or amounts of hemin, than the mutant or the mock pup. It's important to measure the hemin because they wanted to see if iron was present, and iron is essential for bacterial metabolism. So it's essential for the Vibrio cholerae to metabolize. And it was found that metabolic pathways were the highest upregulated genes in Vibrio cholerae during the colonization of the ileum. You could see that there is more iron in the ileum than in the cecum. And having more iron can result in better Vibrio cholerae life. You can see that cholera toxin does, like I said before, enhance metabolism in Vibrio cholerae, according to this study. I also wanted to talk more about how Vibrio cholerae actually infects the body. I know I spoke about this in my previous presentation, but I wanted to go more in depth about it. So basically, it starts with Vibrio cholerae, or whatever you are drinking or consuming that has Vibrio cholerae in it, how it enters the body. So when you consume it, it enters the body and goes to your stomach, and it passes through your stomach because it can survive through the acidic environment of your stomach, and it goes and settles in the small intestines. So once it's in the small intestine, it settles in the lumen, and it starts producing a whole bunch of cholera toxin. The cholera toxin then binds to the GM1 ganglioside, or the B subunit of the cholera toxin binds, and it then goes into the epithelial cell through endocytosis. And once it goes inside the cell, the A subunit of the cholera toxin binds to the G protein coupled receptor. And because the adenylate cyclase is being hyperactivated, it starts producing a lot of CAMP because it can't turn off. So this results in the activation of the CFTR, which is the cystic fibrosis transmembrane conductance regulator, and it pumps a whole bunch of chloride ions into the lumen. This is what leads to the lumen being, or the intestine being filled with liquid and water, and what causes the watery diarrhea. So there are an excess amount of chloride ions being produced. I wanted to see more about what could possibly neutralize it, because the goal is to prevent from the cholera toxin to bind and prevent the symptoms from happening. So a study that I researched was a study that did exactly this. It wanted to figure out a new way to neutralize cholera toxin, and it used nanotechnology-based strategy for cholera toxin neutralization. And they did this by making a nanoparticle solution and a G1 ganglioside solution that they dissolved in water. And adding these two things together, they made a GM1 nanoparticle solution. And this was used as a decoy for the toxins. So it had all the GM1 receptors, but it did not let it bind to the intestinal epithelial cells. So you could see that the goal was for the cholera toxins to bind to this decoy instead of binding to the actual receptors, halting the process that would make the symptoms occur. So continuing on with their experiment, you could see that the results showed that they were able to achieve this. Looking closer into the graphs, you could see that in graph A, they used a fixed concentration of cholera toxin, and they combined it with increasing amounts of the nanoparticles in the solution. And they measured the amount of CAMP that was produced. And with what I discussed beforehand, that this correlates to the amount of cholera toxin that is binding. They compared the results from a PEGNP, which was the control. It was a solution that was of just the nanoparticles and not the GM1 receptor. So there was no binding expected from this. So there shouldn't have been any things preventing the cholera toxin from binding. But the GM1NP solution was the decoy solution that they used. In the beginning, you can see that the nanoparticles amount was really small, so there was little to no effect. But as you increase the amount of nanoparticles in the solution, the amount of CAMP produced was decreasing, so showing that the GM1NP solution did have some effect in preventing the cholera toxin from binding to the actual receptors. In graph B, you can see that they used a fixed amount of the indicated nanoparticles instead and combined that with increasing concentrations of cholera toxin, and they measured the CAMP amounts as well. But since there was a fixed amount of nanoparticles, as the cholera toxins increased, so did the amount of CAMP because there weren't as many receptors for the cholera toxin to bind, so it continued to bind to the regular receptors, the actual ones, and it produced, leading to more production of CAMP. In the third graph C, you can see that they measured the amount of CAMP with an unaffected sample to an infected sample. In the unaffected sample, you can see that there were no CAMP produced, but in the infected sample, you can see that the PEG produced a lot of CAMP, but the GAM1, although producing some CAMP, it was a lot less than the control. So it did prevent from a lot of cholera toxins to bind to the actual receptors and reduce the amount of CAMP being produced. So using this data and information, you can see how the nanoparticles did have a neutralizing effect on cholera toxin. This brings the question of what else has an effect on cholera toxin or on Vibrio cholerae. And bicarbonate has been seen in the process of the infection. And this specific study wanted to focus on its effect on virulence gene expression. In this experiment, they used the two biotypes of O1. O1 Vibrio cholerae is one of the serogroups that creates or produces cholera toxin, like I mentioned before. In experiment 1, they used L-Tor, and experiment C, they used classical strain. And they created a mutant and a wild type, which is the L-Tor or the classical strain, with bicarbonate and with bicarbonate and without bicarbonate. And they grew these for a specific amount of time and measured the amount of cholera toxin that was produced. And you can clearly see that the mutant, which wasn't supposed to have any cholera toxin growth, didn't have any. The wild type without bicarbonate had little to none, and the wild type that had bicarbonate had a lot of cholera toxin growth. And you can also see this in the C graph with the classical strain. You can see that there is a big difference of amount of cholera toxin production. This data does demonstrate that bicarbonate stimulates virulence gene expression by enhancing TOXT activity. TOXT, if I'm saying that correctly, is a regulatory protein that directly activates transcription of genes encoding cholera toxin. So it is the reason for the production of cholera toxin. You can see how bicarbonate does affect the amount of cholera toxin that is being produced. And that is all that I have for you guys. Thank you so much for watching and listening to this presentation.